Hello everyone. This video is going to be trying to answer the question, is image analysis with deep learning ready for mainstream use? Here's some disclosures. All right, so the main takeaways from this talk are going to be that we should monitor data for shifts. Models can cheat, so we just need to be aware of how and how to detect it. Super resolution in image translation can have some issues, specifically hallucination. So we should be aware of those limitations. And then the path forward is likely an AI and physician symbiosis, which I'll explain in this talk. Okay, so first let's talk about issues with data, specifically out of distribution images. So in this use case we're going to talk about a frontal chest X-ray model, which is going to be making predictions from frontal chest X-rays. With that, that model should not be applied to pictures of cats or knee X-rays, right? So these would be considered out of distribution for that specific model, right? And this is pretty straightforward with this use case 1 here, but it becomes more complicated as we look at use case 2, incorrectly acquired images. So here, there's a subtlety of that the user may expect that the model can operate on these images, because the lateral view may contain the information which could be used, but the model is not trained to be able to predict from that specific view. And you can also have pre-processing issues with data, which maybe change the pixel normalization, and this would be something that humans can't detect because the image looks right, but the model doesn't understand that image normalization at all, so it's just going to make erroneous predictions from that, possibly. And the third and most difficult type of out of distribution to detect is samples that are outside of the selection bias. And the selection bias is the samples that are taken for the training and validation data. In this case, adults without pacemakers, right? So images with pacemakers would be outside of the selection bias, and we don't know how well the model works on them because we didn't validate the model on those types of images. You can also expect this would happen with age. If you take a model that's trained on adults and apply it to pediatric images, it may work, but it's not validated, and so it may provide erroneous predictions. And here's an example of how image contrast can actually change the prediction of the model. And so here we change the exposure of the image, and we run each exposure setting through a neural network, and the predictions are shown here. And we can see that they're all kind of changing dramatically. Infiltration is changing very dramatically from 3% to 60% in predicting that pathology. So the image hasn't changed, just some kind of exposure setting on it. And so we wouldn't expect this to happen. Now this neural network wasn't trained to be robust to this. Possibly it could be, but then there can be other artifacts that are modifying the data that could cause other issues similar to this. So it's something we need to be aware of, that these changes can change the prediction for no reasonable reason. Okay, and so let's say we've considered that, and we deploy a system, and we'll call this day 1. We've had, all the technicians and researchers and radiologists have been working on making sure the system works and is deployed and is solving the problems correctly, and that's day 1. Everyone's looking at it. And over time, let's say like a month later, or just the second day, day 2, the data that was used to validate the model, right, this live data, has moved away from the data that it's seeing. It should be the other way. The data that it's seeing has moved away from the data that we validated it on, right? And we really don't know when this is happening, right? So if we start our performance evaluations and we look at the accuracy of some model, when we're about to deploy it, we don't know what the performance will be in the future. So the question is, when do we revalidate this model? And revalidation can be, you take a bunch of patients, maybe new patients, maybe some retrospective study with old patients, and you run them through some system, and then you have radiologists actually score the performance of the model applied to those new images. And so it becomes an issue because do you do that every week, every month, twice a year, right? How can we know when to expect some change in the data, right? Because we're kind of working blind here, and we could revalidate after the model has had lower performance for possibly weeks, right? And so we could prompt changes. Maybe if we know that we have a new piece of equipment in, we then do a thorough revalidation. New diseases, or we look at the patient records and see when they're shifting. We can also look at whenever software updates are happening to any piece of software in the entire chain, from how we're storing it to how we're acquiring it. These can all cause these artifacts to be introduced. Okay, so are there methods to detect these out of distribution examples? Yes, for the use case 1, which is cats, use case 2, which is like a lateral view or an incorrectly formatted image, we have methods that will detect this very well. The use case 3, which is just like pediatric images, this is very hard to detect, and even state-of-the-art methods as of now don't do a good job at detecting this very subtle shift in the distribution. So we just need to be aware of that. Right? And do commercial software vendors employ these safety measures? I asked a CEO of a medical AI software company in 2021, and their response was that their software does not have these issues, it's very robust, which did not instill much confidence in me. So I think as consumers of this software, we need to be aware of what safeguards are built in, and then if they're not built in, look for alternatives to augment and improve that system to make it safer. Okay, so now let's talk about issues with models. And I'm going to highlight this issue called the false positive prediction problem. And in this case, we have a classifier, again, working on just frontal chest x-rays, and it's going to predict the probability of a mass. Okay, so how confident are you in this model's prediction? So here, with this picture shown, the model predicts a mass with 80%. Okay, how about this image? So it's a lateral view. You predict a mass, 80%. Maybe that's more reasonable. Here is a frontal chest x-ray. We predict a mass with 80%, and we can kind of see the mass in the image, so it seems very, maybe likely that that's right. Okay, so here's the source code for this model. I was actually completely ignoring the image. It's not a neural network at all. It's just predicting 80% all the time, right? But somehow we felt like we could trust it more when the image looked more reasonable, right? So this trust can be dangerous. What if the model's wrong, but we think it's right? So how are we safeguarding against this type of situation? And so to answer that question, let's think about how models are flawed in the first place. Often there's going to be an issue where the model is learning via correlation, right? I mean, all models are going to learn via correlation. Just hopefully they find the right correlation, or the correlation we would like it to predict from. This can also be called shortcut learning if we're using the wrong features, right? And we can see this, you can predict shark attacks via ice cream sales. So this issue is not new. It can just become harder to spot with these models. All right, so let's talk about an example where we actually see this with chest x-ray. So here we have two different views of chest x-ray. So we have a PA, where the patient's often standing, and an AP, which the patient is often laying down. All right, depends on the hospital protocol. But they're usually laying down for this view because they're sick, because they have something, they have some issues which prevent them from standing up. And if we look at the average of these images over thousands of patients in these data sets, we can see that there is a difference between these two views, and you can see the difference visualized here. And this is a very easy feature to detect for a neural network because it's so obvious, such a big difference in the image. So if a view is correlated with a pathology, the model can actually just look at the view and then infer the pathology from that. And in this case, where someone's laying down because they can't stand up, they likely have some issues, possibly like pneumonia, and you'd like to predict whether they have pneumonia. So you can just say, the model can say, is the patient laying down? Okay, well, I'll predict a higher probability of pneumonia because that's more likely. Even though that's not the kind of answer we want from the network. Okay. So what are some other shortcuts? So let's say you train a model on multiple hospitals. The equipment between those hospitals could be encoding some information very subtly in the image, and then the model can use that to make its prediction. You can also have machines in different parts of the hospitals. Maybe one is closer to the ICU and the other one is not. And then you get, just between those two X-ray machines in the same hospital, some information that's there that's correlated with the certain types of pathologies that the patients will have. And then we can also look at pathology correlation, right? So if two diseases have a high correlation, and one is very easy to see, and then the other one is very difficult, the model will likely just look at the one that's easier to see, and then extrapolate and also predict that other pathology, right? So this can be kind of deceiving, because if we evaluate it on the data that's sampled from our hospital data, it'll have this correlation too, and we'll think we have really good performance, when in fact for one of them it's maybe not as good as we think. And so can we spot this correlation? Does it exist already? So here's two data sets, one from Spain and one from California, and we can see that there is some correlation between pathologies in just those two data sets. So something to watch out for. Robust evaluations are a key here for identifying this. Okay, so now I'm going to talk about attribution and saliency maps, because they are useful for detecting this type of issue. And so here we see a chest X-ray. We've tried to predict something. I think it was emphysema, but that's not really important here. We plot a saliency map, and then as a practitioner of neural networks, I would look at this image and say, oh, there's a bunch of stuff wrong with this network. So first we can see that there's some saliency at the neck portion. And here, why are we predicting this at the neck? Also, we have some saliency overlapping the patient, some text that's written on the chest X-ray. So we shouldn't be the network shouldn't be looking at that at all. We can also see that there's some saliency being given to the little probe that's attached to the patient. So that's not what we want it to be looking at. And also we can see that it's looking at the side of the patient, right? Likely maybe checking the patient's weight to see if that could be a feature indicating some health conditions as well. And so it's good to get an indication here, saying what the model is using to predict, but I also want to provide some insight into what these saliency maps are doing, because I think there's a big misunderstanding in what they're, how good these are at giving us insight into what the model is predicting. And the way we should think about them is how much a pixel is influential to the prediction. So not explaining why the prediction was made, but just talking about that individual pixel's contribution to the entire prediction. And I'm going to talk about that more. So these things have limitations, as I alluded to. In this example of this dog, picture of a dog, we feed it to a neural network and it predicts dog. And then we compute a saliency map, right, shown here, right to the right of it. And And then what we can do is change that image very subtly using an adversarial attack, which is not really relevant here, just to say that we're changing a few pixels somewhere in the image, and that is actually going to make the model not predict a dog when it sees this exact image in that exact setting, right? But if we generate a saliency map again, we can hardly tell the difference because so many of the pixels are still being used the same way they were before, but we can't even notice the ones that were changed. So that saliency map is looks like it's looking at the dog to predict whatever it's predicting, but it's not predicting what we expect it to predict, right? It's kind of not telling us the truth. It's not giving us the explanation that we want, because it's only talking about individual pixel change. And so I like to say that these saliency maps have kind of been kind of sold by car salesmen to users of these networks, that they can actually do way more than they can. That they can go as far as to explain a prediction instead of kind of the really rudimentary tool that they are, which is just explaining individual pixel change. So I have an example to kind of drive this home further. Here, we're going to take an image of a cat, and we're going to generate an attribution map in a very simplistic way. Here we're going to change the pixel, right? And this is essentially what these image gradient-based methods are doing to generate this attribution map, but I feel like this is much more intuitive. Here we're going to move the pixel in the upper left-hand corner, and then we run that image through the model, right, with all the pixels that we had changed, all the different magnitudes of that pixel. And we see that it changes it just a little bit, right? So here it's moving just 1% down whenever we change that pixel. And then we're going to plot that as the attribution in our attribution map, right? So that pixel is not really influential. And then we're going to do that again with the pixel that's in the cat's ear, right? And we feed that to the model. We see a huge change in the prediction, right? And that's going to drive the pixel in the attribution map to be much higher, right? So I think this is the model we should have when we think about these. This is it's very useful to find spurious correlations, but it's not sufficient to explain a prediction. All right, so what can explain predictions? Or can be useful to address this false positive prediction problem even further. All right, and here I'm going to talk about counterfactuals. So here we have a model that's predicted the mass, whether a patient has a mass, with 80%. We can take a look using the image gradient method here, and we see that there's a whole bunch of attribution around the area of this mass, and that's okay. We can get some confidence, but it's also highlighting other areas. It makes us a little unsure. And then using this counterfactual generation approach, we actually simulate the patient without the pathology. So without having the mass, what would they look like? And in effect, what the image we get is the image with the mass removed, which tells us that what was being used to predict was a mass, right? That gives us a lot of confidence that this prediction was correct. And we're using our knowledge of anatomy to know and kind of check that model's prediction. Here we can look at another example where a model again predicts a mass with 80% probability, but is it right? All right, so we look at the gradient attribution map, and we see kind of attention everywhere. It's kind of got saliency all over the image. But if we look at the counterfactual, the model is trying to remove what it thinks is the mass, right? And it's moving around some of the anatomy, but it looks like it's focusing on this port that's on the patient, right? And so that can lead us to doubt. Maybe this patient doesn't have a mass. This is looking at something that doesn't really make any sense. And the ground truth for this image, as annotated by our radiologist, says that there's no mass in this image. All right, so with these approaches, specifically counterfactuals, we can move towards this physician-AI symbiosis, right, where the models are conveying their predictions graphically, and then physicians utilize their knowledge to kind of detect whether the predictions were incorrect. And when I say graphically, that can be using these counterfactuals or just segmentations in general, where some areas of the image are highlighted and then that's indicating some area to look and verify that something's there. Great. And so now, the next section, we're going to talk about flawed applications. And so here, I'm going to look at these two. So first, there's an approach to convert MRI images to CT images in order to avoid having to perform a CT of a patient. And then we have synthesized H&E staining for microscopy slides, so we can get an image without having to actually stain the tissue. And I want to make sure everyone's thinking about how can these tools be safe for general interpretation. So in that case, right now you look at you get an MRI done, and that image you can read anything you want from that image, because it's the way it's made for general interpretation. You can find indications of anything going on in that image, because you can expect that it's right, that it's going to be able to represent and show you any sort of pathology that could be present in that patient. Right? But when we do this transformation, or this can also be considered super resolution, where is this extra information coming from that's going to be showing us something in a different modality? All right, so I want you to have that in mind, because if a CT didn't provide extra information, why would we have it, right? And same thing for cell staining. Why would we stain the cells if it wasn't showing us extra information that wasn't visible already? Okay, so the way to think about this is even with super resolution, you're going from a low resolution image, you're translating that into a high resolution image, right? And so here we have some source data and target data. And so the source data is lowercase, a few letters of lowercase, and the target data is a few letters of uppercase. And for A, B, and C, we have a really good correspondence between these two domains, or these two data sets. And then in the source domain, we have this element D, right? And that could represent a specific disease or a demographic, right? And then D doesn't exist in our target data. It's missing, right? And instead, we have E and F. So what do we do with D, right? So these approaches, they'll take everything from the source domain and transform it into the target domain, but just the target domain, not extra. So this D will either become E or F, or be grouped in with C and kind of merged in there, right? Because and maybe something else needs to go to E and F. So maybe E and F are going to be hallucinated in that target data in order to properly generate that target data. All right, so with this, just having this imbalance just a little bit, if that model is wrong, you lose all the integrity of that transformation. So I think we should think about this as, it's a square peg in a round hole, and these models are just hammering it in there, right? Because they're so powerful. They can transform anything to anything, right? And potentially erase pathologies or add pathologies. All right, so the things that make this so hard is that it's impossible to balance all pathologies. Once you've accounted for 100, there's probably 101 that you haven't accounted in your tool, and that in itself means it's probably missing something for general interpretation, right? So if you want to conclude things outside of the set of pathologies that you balance between these two data sets. Another issue is that these models are unconstrained. They're just so powerful. They can convert, they can add in tumors into images, as was shown in some paper, or remove tumors from an image, just because it has to map between those two domains, so it's going to do whatever is necessary to make that work. And another issue is, let's say you balance based on all pathologies. They change over time. There are new diseases, and those aren't going to be in the tool that you're using. And when these are deployed, they're only projecting into their target domain. So new data is still going to be projected into the original target domain. All right, so this is makes this very difficult. So my recommendation here is that if we're ever looking at these images, we should verify the source image before making any medical decision, because the source image is coming directly through some known process that we are more familiar with its artifacts. With a vanilla MRI image or CT image, we can draw the conclusion from what's visible there to make sure nothing was added or removed in whatever modified image. And another example here is this fast MRI challenge, which is the idea of this challenge is to take undersampled MRI images, so an undersampled case space, right? So the MRI acquisition time can be cut in half, is the dream of this project. But if you took just that undersampled image and reconstructed it, it would be blurry, right? And that's not what we want. So we'd not like, you can't really see a lot of the details you need to in this blurry image. So the idea here is to operate on this undersampled space and to perform some super resolution task and then reconstruct, have a high resolution reconstruction. And from that high resolution reconstruction, you can do whatever you need to do. Or the argument for this work is to actually use that as a general purpose image for general interpretation. So if we've taken this undersampled MRI, and the only authoritative image we have is this blurry MRI, is that enough to confirm a diagnosis made from the higher resolution image? Possibly. Although looking at this high resolution image, is that going to mislead us and bias us into believing something's there when we look back to the source image, right? So I think we just should be aware of these questions. And here's some recent research studying the errors from these models, and we can see that in this image, the ground truth is shown on the left. It does not have a little fold there, but the reconstructed image has inserted a fold, likely making it more consistent with the data that it's trained on, but not the actual ground truth image that it was trying to reconstruct, right? So we should be aware of these issues. Okay, so now let's talk about what is ready. And the FDA has been approving a lot of these tools, largely in CT and MR. So this is very exciting under the idea that the classification of a software as a medical device. One issue with these is that the 510K submissions to the FDA are private. So we can't look at how these tools were validated, right? We can only see the information that's shared by the vendor of these products. And to further compound this, most of these tools use retrospective studies, right? Looking at historical data. So they're not performing the clinical trials, which would be registered publicly, that we could see. So we can't even look at clinical trials related to the use of that tool. So it's really hard to get a lot of visibility into how these evaluations were done to be able to, try to poke holes in those processes. So that's unfortunate. But we can see these in published research papers, which is very exciting. So one of the applications that I'm excited about that I want to share is this task of pathology segmentation, right? So here we're taking some large histology slide and we're going to identify maybe cells or regions of the tissue that fit some sort of criteria that we're looking for. What's great about this is it's interpretable by default, right? We don't have to go generate counterfactuals or look at salience maps to kind of get some interpretation. It's just that we're actually highlighting the cells that we want to find, right? And then that can be manually verified, right? And then these tasks augment expert users, right? So there's an expert that could look at this entire image, but it's just so much labor to scan this entire slide. But these tools can do it. These tools are able to process these entire slides quickly. Right now, humans are actually not doing this task. So they're just like forming small boxes and then counting in there or doing some analysis in there and then kind of randomly sampling small regions and then from that extrapolating what the whole tissue would have. So this gives us not the best answer, right? So with these augmentation tools, with these tools to process this large slide in its entirety, we can actually get better answers than are what's available now, right? And the logistics of these kind of tools are really nice because the image can be acquired, scanned on site, and then digitally sent through the internet to some processing center. And then the results can be sent back as an image for interpretation there. And then when the image is processed off-site, there can be a quality control that's administered by that vendor to actually make sure that like what's being shown on the image is correct, to kind of make sure that it's done as a service, right? But also, this process can be done locally if desired, right? So that's really flexible in how this tool can work. And an example to highlight this is this OncoPetNet, right, which is a deployed system for mitotic cell counting, which was published last year. And they provide really awesome results. So they're processing over 1,000 slides a day with the system. And if we look at the compute time of this, the basic kind of processing time is about 6.5 minutes. And that's on a single computer. So we can scale up they can scale that up faster to run on multiple machines at once. This is going to cost more, but they can process the whole slide in less than a minute. So phenomenal scale happening with that right now. And the second application I'm excited to share is this acquisition quality assessment. So we have some imaging process that we'd like to not repeat as many times. We would not like to repeat this process many times. And when that happens, there's someone there to check whether the quality is okay, right? And potentially this could be an AI tool. Or that user is using an AI tool to verify their own quality assessment. And then if the quality is not okay, they will reacquire the image, right? But if it's okay, deemed by them, they will send it off for diagnostic interpretation. And at that point, the patient is going to exit a machine, and it's more work, more money to get them back into a machine after. So we'd like to only image them once, right? Because we want to avoid having the radiologist ask for the patient to be sent back in to reacquire the image. All right, so with this setup, having the AI do this quality assessment, we allow non-experts to acquire diagnostic quality images, which is pretty great. In that role, they can process more patients faster because they get this like second opinion about is this image diagnostic quality, right? So they can actually go through more patients. And there's like almost no patient risk here. They come in again. Sure, we want to have them imaged multiple times, but if there's anything going on haywire with this, there's an operator who's determining this, right, whether the quality is okay, and the radiologist who's performing the diagnostic interpretation will see that many images were captured to kind of catch any malfunctioning system right away. So for every time it's used, there is someone looking at the predictions, the result of this model. So it seems like a nice win there. So an example to highlight here is a study on ankle radiographs from Lübeck, Germany. And the algorithm that they developed has a performance of 93% accuracy, where the radiologists are around the same. And the radiographers who are acquiring the images are lower than that, so around 87%. And what they conclude from this study is that for every 100 radiographs, that they could have reduced the number of additional appointments from 13 to 3. Right? So that's just a huge win. All right, so this is a very exciting application that has a lot of potential. Okay, so to recap the takeaways, so we want to monitor data for shifts and be aware over time things could change. So we need to be aware that the system can have those flaws, right? The models themselves can cheat, right? So we need to be aware of that so we can try to spot when that's happening to make sure that we're running this model in a safe way. Super resolution and image-to-image translation is potentially not very safe because it can hallucinate or remove image features from the images. And that I think the path forward is this is an AI and physician symbiosis. And with that, thank you.